Okay, good afternoon, friends. Yesterday we discussed about photosynthesis, wherein we concentrated on first phase of photosynthesis, that is light phase. In that we have seen the structure of chloroplast and the lamellae are the thylakoids, grana thylakoids, stroma thylakoids, and we have seen four important multi-protein complexes that are involved in light reaction. Various pigments such as chlorophylls, carotenoids, xanthophylls, and their location on the surface of the thylakoid membrane. These four complexes were photosystem 2, cytochrome BF complex, photosystem 1, and ATP synthase. We have seen how electrons and protons make a move after absorbing photons of light. First, photosystem 2 becomes activated and ejects its electrons to pheophytin. From there, it moves to quinone, then plastoquinone. Plastoquinone becomes plastoquinol. Then from the plastoquinol, it again gets oxidized to plastoquinone and liberates its protons into the lumen. Electrons are taken up by photosystem or plastocyanin, and then from plastocyanin it moves to photosystem 1, and then again they move to what we call ferredoxin, and ferredoxin in the presence of NADP reductase, this ferredoxin takes protons from stroma and produces what we call coenzyme, reduced form of coenzyme, that is NADPH2. So this is one product, and with the photolysis of water and also with the oxidation of quinol, you see four protons getting liberated, and these four protons are seen getting deposited in the lumen of the thylakoid, where if you compare the concentration of the protons in the thylakoid lumen and in the stroma, so in the thylakoid membrane or in the lumen, more than 1000 times proton concentration is more when compared to stroma. So protons try to escape through mode of diffusion from higher concentration to lower concentration, but thylakoid membrane does not allow them. So here comes the important role played by ATP synthase, which transports electrons one by one. Thereby, if you take the structure of this ATP synthase, otherwise called a coupling factor, which has a stalk and a head. So the protons move from the tail to head, and with the movement of four protons, and that energized protons is sufficient to convert ADP into one ATP. So like that, synthesis of NADPH2 and ATP takes place at the end of the light reaction. And this resulting ATP and NADPH2s are called assimilatory power. This comes to an end of light phase, and these NADPH2 and ATP are called assimilatory power, and this assimilatory power is ultimately used in carbon dioxide reduction. The enzymes for carbon dioxide reduction are present in the stroma. We have seen the enzyme systems for light reactions occur in the grana part, whereas the enzymes for carbon reduction are present in the stroma part. So the end products of light reaction, that is the assimilatory power, is utilized in the reduction of carbon dioxide into carbohydrate. This is what we call dark reaction. Doesn't dark reaction, don't get confused. It is not at all anything to do with the dark or darkness, but it operates independent of light. So the end products of light phase are used in the reduction of carbon dioxide into carbohydrate. Hence, it is called carbon reduction. Now, these enzymes for carbon reduction are present in the stroma. Now, how actually the reduction of carbon dioxide takes place in the stroma, we will learn today. Now, this carbon dioxide reduction takes place in the stroma. Now, there are what we call carbon dioxide acceptors. See, the first carbon dioxide acceptor, based on the nature of the carbon dioxide acceptor and based on the nature of the first stable product formed, plants are classified into three main categories. One is what we call C3 plants, C4 plants, and CAM plants, otherwise called crassulaceous acid metabolism plants. In all these types, ultimately the carbon dioxide gets reduced to carbohydrate. But based on the carbon dioxide acceptor and the enzyme systems and the morphology and the anatomical differences, plants are classified. Say in most or more, almost 80 to 90% of the plants, the carbon dioxide acceptor is RUBP, a 5-carbon compound, rubulose 1,5 bisphosphate. This is the carbon dioxide acceptor, which takes carbon dioxide and ultimately produces an unstable 6-carbon compound, which immediately in a fraction of a second, it dissociates into a stable 3-carbon compound called phosphoglyceric acid, which is a 3-carbon compound. So in all plants where the carbon dioxide acceptor is RUBP and the end product or the first stable product formed is phosphoglyceric acid, such plants are called C3 plants. C means carbon, first stable product is phosphoglyceric acid. Hence, they are called C3 plants. And the mechanism, all the biochemical reactions that take place in this mode of carbon dioxide fixation were elucidated by the exemplary works of Melvin Calvin and his associates. And for their exemplary work, they were awarded Nobel Prize in 1961. So as this detailed, biochemical reactions that take place in a cyclic fashion, hence it is called a C3 cycle, as these were elucidated by the famous scientist Calvin. Hence, this cycle is also named after him as C3 cycle. And the plants where this mode of carbon fixation occurs are called C3 plants. Now, the second category is, of course, we'll study them in detail. Second category is C4 plants. So, till 1970s, people thought that all plants do carbon dioxide fixation by Melvin Calvin pathway or C3 pathway. But in later on in 1970s, especially it was in 1965, quote shock. Later on, Hatch and Slack, scientists from Australian University, they were amazed to see that in sugarcane, they saw that the first stable product formed was a 4-carbon compound, that is oxaloacetic acid. And the carbon dioxide acceptor was a 3-carbon compound, phosphoenol pyruvate. So here the carbon dioxide acceptor is phosphoenol pyruvate and the stable product formed is oxaloacetic acid, a 4-carbon compound. Hence, this cycle is called C4 cycle. Oxaloacetic acid is a C4 cycle, C4 carbon compound. Phosphoglyceric acid is a C3 compound, whereas oxaloacetic acid or aspartic acid or malic acid, these are all intermediaries, first stable products. As it is a first stable product is oxaloacetic acid, a 4-carbon compound. Hence, the plants where the first stable compound is oxaloacetic acid, such plants are called C4 plants. And as it was discovered by Hatch and Slack, hence it is also otherwise called Hatch and Slack cycle. So Hatch and Slack cycle or C4 cycle is one and the same. Later on, In almost all the plants, stomata open during daytime, close by evening. But there is a special type of plants, especially xerophytic plants, especially plants belonging to Crassulaceae. For example, our Bryophyllum. In Bryophyllum and other Crassulacean members, the stomata open during night time. They close during daytime. Why? Because these are xerophytes. There, the water availability is very less. To conserve whatever the moisture that is present in them, they have adopted a mechanism wherein stomata remain closed during daytime and they get opened during night time. And this mode of fixation of carbon dioxide during the night is found only in crassulacean members. Hence, this mode of fixation is called CAM pathway, crassulacean acid metabolism pathway. So these are three major types of carbon dioxide fixations. Now we'll see in detail one after the other. First of all, we'll begin with C3 plants. Now C3 plants, as I already told you, the C3 cycle. Why it is known as C3 cycle means here the first stable product formed is a 3-carbon compound, that is phosphoglyceric acid. So for our convenience, the entire cycle or C3 cycle can be divided into 3 major steps. One is what we call carboxylation, second is called reduction, and the third is called regeneration. So first one is carboxylation. What do we mean by carboxylation? Carboxylation means taking of carbon dioxide or fixing of carbon dioxide is the first step. So first carbon acceptor is a 5-carbon compound by name RUBP, rubulose 1,5 bisphosphate, a 5-carbon compound, takes carbon dioxide, and in the presence of RUBP carboxylase. RUBP carboxylase is the enzyme that determines this biochemical, or that regulates this carbon fixation or carboxylation. So for our convenience, so for example, if you start with 1 carbon dioxide, for the formation of 1 glucose molecule, the cycle has to repeat 6 times. When it repeats 6 times, 6 carbon dioxide molecules will ultimately get converted into one glucose molecule. So for our convenience, we start with, we take 6 molecules of RUBP. So 6 molecules of RUBP take 6 carbon dioxide molecules and produce 12 molecules of phosphoglyceric acid. See, RUBP, I told you, a 5-carbon compound. By taking carbon dioxide, 5 becomes 6. Now this 6-carbon unstable product immediately splits into 2 3-carbon phosphoglyceric acids. That is a stable product. As it is a 3-carbon compound, hence we call it as a C3 cycle or Calvin cycle. So first step, 6 carbon dioxide molecules taken by 6 molecules of RUBP in the presence of RUBP carboxylase enzyme and produces 12 molecules of phosphoglyceric acid. Now these 12 molecules of phosphoglyceric acid take 12 ATP molecules. Remember, ATP and NADPH2 are the assimilatory power produced in light phase. So one end product, that is ATP, will be taken up here by 12 molecules of phosphoglyceric acid, take 12 molecules of ATP and get converted into what we call 12 molecules of glycerol 1,5 phosphoglyceric acid. So diphosphate, otherwise called, first is phosphoglyceric acid. By taking one more, it becomes diphosphoglyceric acid or bisphosphoglyceric acid. Now this 1,5 bisphosphoglyceric acid, now this or 1,3 bisphosphoglyceric acid, in the next step, it gets reduced by using NADPH2. So glyceric acid. By taking NADP, it becomes phosphoglyceraldehyde. Glyceric acid gets reduced to glyceraldehyde. So actually, with the formation of glyceraldehyde, almost fixation of carbon dioxide is over. From hereafter, so we have seen utilization of ATP in the first phase. In the second stage, NADP was utilized. So both the assimilatory powers are utilized. First, in the phosphatization of RUBP. In the second, phosphoglyceric acid gets reduced to phosphoglyceraldehyde. Now, of these glyceraldehydes, the glyceraldehyde 3-phosphate, then it changes to its isomer in the presence of triose phosphate isomerase into its isomer called dihydroxyacetone phosphate. So glyceraldehyde phosphate and dihydroxyacetone phosphate, these two are isomers. Both of them are 3-carbon compounds. Now glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, these two combine in the presence of an enzyme, aldolase enzyme, and produce 2 molecules of fructose 1,6 bisphosphate. And these fructose 1,6 bisphosphate, in the next step, it uses glyceraldehyde phosphate and produces 2 molecules of xylulose 5-phosphate and erythrose 4-phosphate. So remember, fructose is a 6-carbon compound. Glyceraldehyde is a 3-carbon compound. 6 plus 3, it becomes a 9-carbon. So 9-carbon compound breaks into a 5-carbon compound and a 4-carbon compound. 5-carbon compound is called xylulose 5-phosphate and 4-carbon compound is called, 4 is erythrose. So xylulose 5-phosphate and erythrose 4-phosphate. And in the next step, erythrose 4-phosphate takes or combines with the dihydroxyacetone phosphate. Erythrose 4-phosphate is a 4-carbon compound. Dihydroxyacetone phosphate is a 3-carbon compound. 4 plus 3 unite in the and form what we call a 7-carbon compound called pseudoheptose 1,7 bisphosphate. In the next step, it loses its phosphate by phosphatase enzyme. It becomes 1,7 bisphosphate becomes pseudoheptose 7-phosphate. Now pseudoheptose 7-phosphate combines. Now pseudoheptose 7-phosphate combines with 2 molecules of pseudoheptose and 2 molecules of glyceraldehyde in the presence of an enzyme transketolase and produce a 5-carbon xylulose 5-phosphate and a 6-carbon ribose 5-phosphate. See, pseudoheptose is a 7-carbon compound. Glyceraldehyde is a 3-carbon. 7 plus 3, 10. It breaks into 2 5-carbon compounds called xylulose 5-phosphate and ribose 5-phosphate. Then xylulose 5-phosphate in the presence of an enzyme epimerase, it becomes ribulose 5-phosphate. And ribose 5-phosphate in the presence of an enzyme, it becomes ribulose. So altogether we get 6 molecules of ribulose 5-phosphates. So we have started with 6 molecules of ribulose. So here there is a regeneration of 6 ribulose 5-phosphates. 6 molecules of ribulose 5-phosphate take 6 molecules of ATP and thus give rise to 6 molecules of ribulose 1,5 bisphosphate. So here this production of again ribulose biphosphate, bisphosphate is called regeneration. So first there was a reduction by taking ATP and ATP and NADH. That is reduction. Then regeneration is, so these 4-carbon, 5-carbon compounds, they fuse, they submerge in the presence of aldolase enzymes and transketolase enzymes, and they convert themselves into a 6-carbon sugar, a 5-carbon sugar, a 7-carbon sugar, a 4-carbon. So like this, through various combinations, ultimately the ribulose 1,5 bisphosphate is regenerated. Hence, it is called regeneration of phase. This is what we call Calvin cycle or C3 cycle. Then we move to what we call C4 cycle or C4 plants. I told you this was in detail worked out by Hatch and Slack. Hence, it is named after them as Hatch and Slack cycle. As the stable product is oxaloacetic acid, it is also called a C4 cycle. Now, here in C4 cycle, generally C4 pathway is restricted to only few plants, especially in Graminae members. In Cyperaceae members, you will come across this C4 method of in Zygophyllaceae members, in certain other members of other dicotyledonous members. Mostly it is restricted to Graminae or Poaceae. Now, as it was first found in sugarcane, it is also called sugarcane pathway or maize pathway. So this C4 pathway, now you have to remember here, this occurs only in certain cereals. Only in certain cereals such as your maize, sorghum or sugarcane, you can come across this, or in tribulus and other members, certain amaranthus members also, you can come across this C4 pathway. Now in C4 pathway, the anatomy is quite different. In C4, of course, you know, it mostly occurs in monocots. In case of dicots, we have seen wherein if you take the anatomy of the leaf, you find, upper epidermis, lower epidermis, and the mesophyll. Mesophyll differentiated into, palisade parenchyma and spongy parenchyma. Of course, in the center, you have vascular bundle. In monocots, the veins, vascular bundles are nothing but veins. The veins are parallel. Now, here in C4 plants, you find a special type of anatomy, called Cronje anatomy. Here, unlike our dicots, the mesophyll cells are uniform as such. And in the center, you find what we call a vascular bundle. The vascular bundle is nothing but xylem and phloem, and they are surrounded by conspicuous layer of cells. This what we call bundle sheath cells. This bundle sheath. So this bundle sheath, if you observe under microscope, it looks like what we call a cronje, that is nothing but a garland. It looks like so in Germany, this cronje means guldasta, what we call a garland. And as it looks like a garland, hence the cronje anatomy. So in the center, that is in the vascular area, xylem and phloem are surrounded by specialized cells called bundle sheath cells. Now in bundle sheath cells, the chloroplasts are big, but they are of A grana type. Whereas in mesophils, you find large number of chloroplasts, but they are small. They are grana. So in monocots, you find two types of chloroplasts. This is what we call dimorphism. Two types of chloroplasts are present. Small and with conspicuous chloroplasts in mesophyll, and large chloroplasts which are of A grana type are present in the bundle sheath cells. Now, this is what we call Cronje anatomy. Now, in C4 plants, in mesophyll, you see C4 cycle, and in the bundle sheath cells, you see C3 cycle. So, in C4 plants, you see both cycles, C3 and C4 cycles, both operate in C4 plants. Hence, C4 plants are highly productive. Their production, photosynthate, is much more than the C3 plants, because in C4 plants, both C4 and C3 cycles operate simultaneously. Hence, the photosynthate produced is more. Now, here the cycle is very simple. Here, you remember both the mesophyll cells and bundle sheath cells participate in the carbon reduction. So first we will begin with mesophyll cells. Mesophils, here, unlike your 5-carbon compound is C3 plants, here the carbon dioxide acceptor is a 3-carbon compound called phosphoenol pyruvate PEP. PEP are the phosphoenol pyruvate. This is a 3-carbon compound. A 3-carbon compound PEP takes in carbon dioxide and produces a 4-carbon compound called oxaloacetic acid. This oxaloacetic acid is quite unstable. It immediately it gives rise to what we call malic acid or aspartic acid. So based on the stable product, malic acid or aspartic acid, plants are different. Say, for example, malate farmers are, for example, in sugarcane and maize, they are called malate farmers, whereas aspartate farmers are, for example, amaranthus and Eleusine coracana. Here, the first stable product is aspartic acid. So, whatever the case may be, so the first stable product is a 4-carbon compound. Hence, they are called C4 plants or like cycle or C4 cycle. So now the carbon dioxide acceptor is PEP, which takes in one bicarbonate molecule and gets converted into oxaloacetic acid. And this oxaloacetic acid changes to malate in mesophyll. And this malate, oxaloacetic acid, gets reduced to malic acid, NADP, malic dehydrogenase. Now this malate immediately moves to bundle sheath cells, wherein the malate gets decarboxylated and dehydrogenated. As a result, so malate is a 4-carbon compound. It loses carbon dioxide in the presence of malic dehydrogenase and decarboxylase and produces a 3-carbon compound called pyruvate, and carbon dioxide is released. And this carbon dioxide that is released in bundle sheath cells will be accepted by C3 enzyme, that is RUBP carboxylase enzyme by RUBP carboxylase, RUBP carboxylase bisphosphate. In the presence of RUBP carboxylase, it takes this carbon dioxide and it repeats what we call C3 cycle. So now here the pyruvate, say oxaloacetic acid, malic acid. From malic acid moves into bundle sheath cells, it gets decarboxylated dehydrogenase. You get release of carbon dioxide and formation of pyruvic acid. This pyruvic acid again moves back into mesophyll cells, where the pyruvic acid takes ATP and forms phosphoenol pyruvate. Again, the cycle gets repeated. So here in bundle sheath cells, C3 cycle is operating. In mesophyll cells, C4 cycle is operating. Hence, they are more efficient as far as carbon dioxide fixation than the C3 plants. Now, they may ask you what we call differences between C3, C4 plants, very important. So in C4 plants, PEP is the carbon dioxide, phosphoenol pyruvate is the acceptor, RUBP is the acceptor there, and PEP carboxylase is the enzyme, RUBP carboxylase is the enzyme, and the first stable product is oxaloacetic acid. And C3 plants, first is phosphoglyceric acid. So now, generally, the photorespiration is absent in C4 plants. Photorespiration is very high in C3 plants. And generally, the yield is more in C4 plants, less in C3 plants. And generally, the temperature optimum of C4 plants is more. They can do photosynthesis even when the temperature is more than 40. But in case of C3 plants, it gets reduced. If you expose C3 plants to temperatures more than 40 degrees centigrade, generally the photosynthate yield gets reduced. So now C4 plants have Cronje anatomy, and Cronje anatomy is absent in C3 plants. So like this, you have to remember. These are the important differences between C3 and C4 plants. Then we move to CAM plants, otherwise called crassulation acid metabolism plants. Here the cycle remains very similar to C4 plants. It is very, very similar to C4 plants, but the difference is in C3 plants, the two cycles, that is C3 and C4 cycles, operate in different cells. That is, in mesophyll cells, C4 cycle, in bundle sheath cells, C3 cycle. But unlike that, in CAM plants, both the cycles, both the types of fixation, C4 fixation and C3 fixation, take place within the same cell, but at different times. I told you here, the stomata open during night time. So during night time, the carbon dioxide fixation takes place through C4 pathway, and by night time, the oxaloacetic acid produced, and this oxaloacetic acid changes to malic acid and it gets stored in the vacuole. And this malic acid during daytime, when stomata close, then the malic acid metabolism takes place. The malic acid gets decarboxylated and dehydrogenated and gives out CO2, and this CO2 will be fixed by C3 enzymes, and pyruvate is produced, and this pyruvate ultimately changes to starch. But only difference is in C4 plants, the pyruvate again takes ATP and becomes phosphoenol pyruvate. But here regeneration is different here. The starch itself produces, by hydrolysis, starch produces trioses, and which is nothing but PEP. So both cycles, C3 and C4 cycle, takes place within the same cell, but the difference is they are temporally separated. That is, one occurs during night time, the other cycle occurs during daytime. This is the difference between these two. Okay, now we learn about what we call photorespiration. You know respiration? Respiration is a process wherein this is operated by the important cell organelle, that is mitochondria. Hence, mitochondria are called powerhouses of the cells. They take, so what happens? The oxidation of glucose in the presence of oxygen, and of course, energy rich oxidative phosphorylation, what we call, wherein ATP is produced and carbon dioxide is produced. That is intake of oxygen and breakdown of glucose into what we call carbon dioxide and energy is called respiration. So as the respiration is occurring in the presence of oxygen, this is called aerobic respiration. So here the organelle involved is what we call mitochondria. But in C3 plants, in addition to normal respiration, when plants are exposed to high temperature, high intensity of light, and more concentration of oxygen and less concentration of carbon dioxide in them, the RUPP carboxylase, instead of doing carboxylation, it acts as RUBP oxygenase. So RUBP, instead of doing carboxylation, it gets oxidized into producing a 3-carbon compound and a 2-carbon compound. A 2-carbon compound is called glycolic acid, and 3-carbon compound is our triose. And of course, this triose, of course, it produces again starch. So now, what is happening here? The RUBP, instead of giving 2 molecules of PGA normally, here it is giving only 1 PGA, and the other 2 carbons are getting modified into glycolic acid in chloroplast, and this glycolic acid moves to peroxisome in the next step, where it breaks into, peroxisome in the glycolic acid changes to, oxidizes to glyoxylate, and glyoxylate changes to glycine. And this glycine moves to what we call mitochondria, where it gets decarboxylated and produces serine, and serine again moves back to peroxisome. And here again, the hydroxypyruvate changes to glycerate, and this glycerate again moves back into chloroplast, where phosphorylation takes place and forms PEP. So here, this is a light-induced respiration, hence it is called photorespiration. Normal respiration takes place during night and daytime, but this respiration, which is light-induced respiration, hence it is called a photorespiration. In addition to normal respiration, this is another mode of respiration occurring in C3 plants. It is a wasteful process. So hence, C3 plants are, as they have this photorespiration, they are less efficient when compared to C4 plants. So this light-induced respiration involving 3 cell organelles, chloroplast, peroxisome, and mitochondria, and the glycolic acid, a 2-carbon compound, gets in a cyclic fashion, hence it is called C2 cycle or photorespiration. Now, last we will learn about what are the various factors that influence rate of photosynthesis. Now you see respiration, this photorespiration, of course, photosynthesis, all are regulated by various environmental factors and also internal factors. So the factors that regulate photosynthesis, for our convenience, we divide them into two. One is what we call internal factors, the other is external factors. External factors means environmental factors such as light, carbon dioxide, temperature, oxygen, water, nutrients, etc. They are all external factors. Light, if light is bright, generally respiration rate will be more. But if it is very severe, in such case, the stomata may close, because lot of evaporation takes place, transpiration takes place. It induces closure of stomata. Hence, photosynthesis drops. So, light, of course, quality of the light and the duration of the light both determine the rate of photosynthesis. Then temperature, of course, temperature for each, starting from 10 to 20 up to 30 degrees centigrade. For every 10 degrees centigrade, there is a doubling of photosynthetic rate. But after 35 or 40 degrees centigrade, there is a sharp fall in the photosynthetic yield, because that we have seen in a light reaction, that is red drop or Emerson enhancement effect. Then, of course, concentration of carbon dioxide is very, very important. If you increase the rate of availability of the carbon dioxide, then photosynthesis increases. But if you increase the concentration of oxygen, there photorespiration takes place and a photosynthetic yield gets diminished. So in addition to this, these are some of the factors. Of course, water is another important factor. Sufficient water should be there. Water is very important. Photolysis. Water is the source for protons. Protons are the source for ATP formation. So water is also another important. So temperature, light, water, carbon dioxide, oxygen, all are very, very important for photosynthesis to occur. Then what are the internal factors? In addition to external factors, even internal factors are also play an important role in the photosynthetic yield. The first is, of course, the pigments, chlorophyll pigments. So generally, if chlorophyll pigments are more, generally it will be more. But if carotenoids are more, generally photosynthetic yield gets reduced. And not only that, even the hormones hormones also play an important role. See, for example, if certain hormones, for example, gibberellins and cytokinins, they are known to promote what we call opening of the stomata. If the stomata open widely, then plant can easily intake carbon dioxide, thereby it enhances photosynthetic yield. So also if hormones, in addition to hormones, even enzymes also play an important role. We have seen if the concentration of oxygen becomes more, photorespiration gets enhanced. RUBP carboxylase, instead of acting as a carboxylase, it functions as an oxygenase. And so photorespiration, it promotes photorespiration. So like this, various internal and external factors determine or regulate rate of photosynthesis. So Blackman's limiting factors. So like photosynthesis, wherein we have seen so many factors, external factors, internal factors regulate photosynthesis. If a biosynthetic pathway is regulated by more than 3 or 4 important regulators, then the rate of that particular reaction will be determined by the factor that is at its lowest. Say, for example, if you increase carbon dioxide, then photosynthetic yield gets increased. But too much increase of carbon dioxide results in closure of stomata. Okay, so if you increase oxygen too much, then instead of carboxylase, RUBP carboxylase, RUBP oxygenase becomes active. So if 2 or 3 factors are there, to certain extent it moves. Later on it becomes stationary, then it falls. What? Because if you are increasing one factor, the other factor becomes limiting factor. So it is the factor that is at its lowest concentration that determines the rate of that particular biosynthetic pathway. So like this is what we call Blackman's limiting factors. So this is about various modes of carbon fixation, C3, C4, CAM, photorespiration, and various factors that determine the rate of photosynthesis we discussed. Now, if you have any doubt regarding this, of course, we had to move a little faster today because a lot of portion is yet to be covered, and we are moving in a prefixed way. So if you have any doubt, you raise your doubts. This 5 minutes for your doubt clearance. So even not only in today's class, but you can also raise questions for the classes that are already over. So if no question, so let us call it a day. Of course, we have one more class tomorrow. We'll be meeting same time from 3 to 4 tomorrow. Until then, bye.